Welcome everyone to the webinar of topics in animal-free innovation and cosmetics and drug safety assessment. Webinar is organized by MDPI, and MDPI is a pioneer in scholarly open access publishing and has supported academic communities since 1996. MDPI is based in Basel, Switzerland, and MDPI has the mission to foster open scientific exchange in all forms across all disciplines. The webinar is organized by two journals, Journal Life and Journal Cosmetics. Journal Life is an international peer-reviewed open access journals of scientific studies related to fundamental themes in life sciences, from basic to applied research, published monthly online by MDPI. Journal Life is indexed in Scopus, Web of Science, PubMed, and many other databases. Journal ranking is currently in 25% of all biology journals. We are also supported by Journal Cosmetics, who is an international scientific peer-reviewed open access journal on the science and technology of cosmetics, published bi-monthly online by MDPI. Journal Cosmetics is highly visible and indexed in Scopus, Web of Science, and many other databases. As you can see, Journal is currently in top 25% of dermatology journals. Here I'm presenting you the webinar agenda and all the speakers that will be sharing their topics on this, or they will be sharing their talks on this topic. First, we'll have the chair introduction from Professor Maria Pilar Vinardel, who will be, after that, we are followed by the first speaker, Dr. Katrin Herrmann, who will be discussing on recent advances in non-animal methods for cosmetics and drug safety assessment. After that, we will be having Professor Dr. Emanuela Corsini, who will be discussing on innovative approaches to skin and photosensitization. After that, we have the Professor Dr. Thomas Hartung, who will be a bit later for this, who will join us a bit later on this webinar, and he will be discussing about the alternatives to animal testing. After that, we are following by the Q&A session, and also after Q&A, we will have the closing of webinar by our chair, Professor Maria Pilar Vinardel. For the Q&A session, please leave your questions in the Q&A box, and then we will try and provide all the answers to your questions. Please, if you would like to have any talk, please raise your hand and we will discuss your questions. And here I would like to present each of the speakers that we will be introducing today. Our webinar chair is Professor Dr. Maria Pilar Vinardel, who is working in the Department of Biochemistry and Physiology, University of Barcelona, Spain. We also have Dr. Katrin Herrmann, who is working on Johns Hopkins University Center for Alternatives to Animal Testing. We also have Professor Dr. Emanuela Corsini, who is working on the Department of Pharmacological and Biomolecular Sciences from University of Milano, Italy. And we have Professor Dr. Thomas Hartung, Department of Environmental Health and Engineering at the Johns Hopkins Bloomberg School of Public Health in the USA. Now we will have our chair, Professor Dr. Maria Pilar, to discuss about the introduction of this webinar. So please, Professor, take your word.
Thank you for your kind presentation. And first of all, welcome everybody. And thank you for joining us today in this interesting webinar about animal-free innovation in cosmetic and drug safety assessment. The more important is that the world of product development is changing rapidly, and regulatory pressure, scientific advancement, and growing public awareness are pushing us toward safer, more ethical, and more efficient methods of testing. In this webinar, we'll explore how the new methodologies are transforming the way we assess the safety of cosmetic and also pharmaceuticals. We'll hear from 3 experts in the field, and who have been present before the 3. I will make a little introduction of the different speakers. I am a pleasure to introduce Dr. Katrin Herrmann. She is a PhD in biomedical science from the Free University of Berlin and veterinary specialist in animal welfare science, ethics, and law. And then for this reason, she will introduce us to this aspect more ethic and about the alternative method. She's a senior associate at the Johns Hopkins University Center for Alternative to Animal Testing, when she directs the Beyond Classical Refinement and Education programs. Through her global advocacy and academic leadership, she advances a more compassionate and scientifically sound vision for the future of research and testing. Her talk is Recent Advances in Non-Animal Method for Cosmetic and Drug Safety Assessment, from scientific innovation to regulatory uptake. Thank you again, Katrin, for your participation, and you can share your presentation to all the audience.
Yeah, thank you for the invitation. I'll show how we can move from innovation in non-animal methods to real regulatory use in cosmetics and drug safety, what's changing in policy, what still slows adoption, and what scientists and regulators can do now already. I will cover recent developments, some decisive, others incremental, and how targeted training and a replacement first mindset can make non-animal methods the default for human relevant decisions. So here's why we need to shift. Our current development pipeline is too often fails when it reaches human beings. Systematic analyses show that more than 92% of candidate medicines that do not succeed in clinical trials, often because safety or efficacy in people is not predicted by the preclinical package. The consequences are serious: risk to volunteers and patients, the use of animals without human benefit, years of lost time, and of course, billions in sunk cost per approval. For laboratory scientists, human relevant approaches reduce false positive and false negative findings and allow for faster decisions to proceed or to stop. And for regulators, these approaches can provide clearer evidence grounded in human biology, which supports more confident and more transparent decisions. Two independent analyses show momentum. Taylor et al. Reviewed 7 biomedical fields across the US, China, France, and Germany and the UK, and they found that by 2022, non-animal only publications outpaced animal papers in several areas, for example, breast cancer and blood cancer and others. And they saw a sharp rise from 2016 to 2019. And the global ratio rose from 1.5 to 2.7. The UK is the leader with 4.5 to 1, and China is a little bit lagging behind, but also growing. US, France, and Germany are about near 3 to 1. And a separate ALTEC study confirms this trend. Within NAMs, human tissue systems and computational models drive most growth, with organ-on-chip platforms expanding quickly and increasingly linked to decision contexts. Barber et al. Note a shift from raw accuracy to true decision support. A quick regulatory win, the OECD test guideline 497, which integrates multiple NAMs for skin sensitization. It integrates what actually the scientific committee on consumer safety notes is that these approaches provide information equivalent to the historical local lymph node assay in mice, and that allows many submissions to replace the legacy animal tests. And next, I will present the global status at a glance, separating reforms, operational pathways, signals, and pending items. So we don't overstate uptake, where it's not yet formalized. This distinction can help scientists prepare dossiers and help regulators set realistic expectations. In the United States, the Food and Drug Administration Modernization Act 2.0 removed the statutory requirement to use animals as the default for drug development. The US Food and Drug Administration has also established a standing pathway to qualify new tools for specific uses, and it has published a roadmap that encourages sponsors to include non-animal evidence and applications to begin clinical studies. In parallel, the US EPA has publicly committed to end the use of mammals for testing by 2035. In the EU, the European Commission has issued a roadmap that aims to phase out animal testing and chemical safety, and committees have started to integrate next-generation risk assessment and integrated approaches to testing and assessment into guideline. The European Medicines Agency work plan prioritizes qualification of non-animal methods. So these are strong signals. The impact now depends on implementation, quality, and pace. In the United Kingdom, the government partially reinstated the ban for ingredients used exclusively in cosmetics, while requirements for worker and environmental protection can still trigger testing under the National Chemicals Framework. A more detailed government position and a roadmap with acceptance criteria are awaited. In Australia, the Industrial Chemicals Introduction Scheme provides pathways for non-animal evidence, including in vitro data, computational models, and weight of evidence reasoning. And I'm ending this overview with the Organization for Economic Cooperation and Development. As more non-animal methods enter OECD test guidelines and next-generation risk assessment work programs, the mutual acceptance of data system ensures that studies performed to a guideline in one member country are accepted by others. And this is how regional progress becomes globally portable. Now we will take a look at the cosmetics animal testing bans, or how far we are developed there in the different areas. And in the EU and European Economic Area, both the testing and the marketing of marketing have been fully in force since 2013. Which means finished cosmetics and cosmetic ingredients tested on animals cannot be sold in the European Union. And this remains the anchor for many jurisdictions. And beyond Europe, Canada brought a national ban into force at the end of 2023, covering both testing and trade. And in 2023, Brazil issued the CONSEA Resolution 58, which limited cosmetics animal tests. But there wasn't a full ban. The full federal ban came later, just recently, on July 30th, actually, covering cosmetics, personal care products, perfumes, and their ingredients. Mexico adopted a national ban back in 2021 that also prohibits manufacturing, import, and marketing of animal-tested cosmetics. And Chile followed with legislation in 2023 that takes effect in 2025 and likewise covers testing and trade. Colombia adopted a ban in 2020 and has strengthened and enforced in 2024 with narrow exceptions defined by law. And these moves show solid momentum across the Americas. And when we look at Asia Pacific and the Middle East, there are multiple countries that have bans or strong restrictions, though the scope varies. In India, India has both a testing ban and an import ban. Israel has a domestic testing ban and a ban on imports and sales. And for example, South Korea restricts the distribution and sale of newly animal tested cosmetics, whereas New Zealand prohibits domestic cosmetics testing. These are not identical frameworks, but together they point in the same direction. Then when we look at the United States, there's a strong state-level action. 12 states have bans on newly animal-tested cosmetics. At the federal level, the Humane Cosmetics Act of 2025 has been introduced in Congress, and if passed, it would set a national standard. And finally, although China does not have a ban since 2021, many imported general cosmetics can qualify for exemptions from animal testing when conditions are met, which is a meaningful partial shift. And the takeaway is here that bans and restrictions are spreading and the direction of travel is clear. And scope and expectations differ by jurisdictions, so dossiers need to be tailored, yet the opportunity is real. Companies that standardize non-animal methods and documentation now will be best positioned in this evolving landscape. This brief divider is simply to signal a pivot from principles to practice. In the next slide, I will highlight specific endpoints and decision contexts where non-animal approaches are already usable or where many of you can move quickly. So there are 5 areas where laboratories and assessors can act now. First, pyrogen testing. The monocyte activation test and recombinant factor C can replace the rabbit pyrogen test. Second, skin and eye irritation and corrosion. They're validated in vitro batteries are available and already used in many regulatory contexts. And third, skin sensitization, the OECD test guideline number 497 describes defined approaches that replace legacy animal assays. And then we have the genotoxicity in specific contexts. In vitro micronucleus test combined with explicit weight of evidence reasoning can answer many questions already. And fifth, and the next generation risk assessment integrated approaches to testing and assessment. These frameworks enable fit-for-purpose decisions without new animal data when the scenario is well defined. So now next are the barriers to adoption that exist. And I want to start with more the psychological and institutional barriers. Maybe let's frame it like that. I want to explain with this why better tools do not automatically lead to rapid adoption. I describe them as the 3 rings or 3 layers that enforce the status quo of animal use in science. There's the personal layer or ring of conditioning, the organizational layer of norms and incentives, and we have the system layer of infrastructure and regulation. And understanding these layers help us design effective interventions. So think of 3 reinforcing rings. The personal conditioning is you're repeatedly exposed to procedures that can dull empathy. I'm talking about academia and where you're trained. Then distress might be often reframed as compassion fatigue instead of a signal to change practice. Then we have the middle ring, the organizational culture and norms. And for example, review committees and peers may ask, where is the animal data? And the economics of animal facilities can reward keeping cages full. And then we have the outer ring, decades of funding and supply chains built around animal models, they are reinforced by academic incentives that favor tradition. And important to know is, of course, each ring strengthens the other, and the change, when we want to change the system, we need to address all the 3 rings at once. Although we see field-wide momentum, universities remain a key bottleneck. Most animal use still happens in universities and medical schools. So what students are taught, what facilities exist, and what publishing norms are enforced largely determines how quickly the field can shift. As I mentioned earlier, there are analyses showing that there is already a clear trend that there are certain biomedical areas where non-animal methods are really gaining ground. The momentum is there, but if we want to accelerate it, we need higher education to catch up. And that means updating curricula to include NAMs, building access to non-animal methods course and platforms, and shifting journal expectations so that students and researchers aren't pressured to produce animal data just to get published. And the implication is clear: if we fix these academic structures, the broader trend away from animal use will move much faster. So just to talk about the Center for Alternatives to Animal Testing's education program, what I just explained, we need to train the next generation of scientists. So to close this gap, we offer 2 MOOCs on Coursera, and maybe Thomas will say something about that too in his talk. He has over 10,000 learners per course, and it's very well received, and we are planning on expanding those MOOCs. And we also have a bi-monthly 3Rs webinar series that has a strong focus on replacement. We also organize early career scientists workshops where we focus on early career scientists' needs. So it's not only about science, but also helping them with grant applications and other things that are important to them. And we start a new series now for alternatives in simulation and education, together with Internis and Ombion. And at the moment, there's a call for microgrants for reduction proposals and humane education proposals. So please check these out. And also important is training, obviously, the regulators and building confidence. So there, it would be good to have joint learning labs, assessors and developers co-review real cases. And then we have to teach context of use, performance standards, uncertainty, weight of evidence. And another important topic, mutual acceptance across agencies, adaptive validation pathways, and also co-review turns possible into probable acceptance. So there are 4 practical levers that institutions can pull today or this year. First, fund the connective tissue standards, validation, qualification networks, and shared data platform so that non-animal approaches become dependable workhorses for regulatory submissions. Second, journals can operationalize fairness by adopting anti-bias checklists and by clearly accepting manuscripts that rely only on non-animal methods when appropriate. Third, procurement can switch to animal-free media and reagents consistent with good cell culture practice. And fourth, leaders can track key performance indicators such as the percentage of projects that use validated non-animal methods, time from submission to decision, and the amount of animal use avoided. What gets measured gets managed. And just a few words on why roadmaps are so worthwhile if they have like certain things in them. Let's see about the EU roadmap that looks very promising. The roadmaps prevent drift, and the new European roadmap proposes harmonized guidelines across sectors such as chemicals, pesticides, medicines, and also worker safety, faster validation and qualification, investment in education and training for both scientists and assessors, and shared data platforms and transparency, so animals are the last resort. And the determining factors now are the quality of implementation and the speed of delivery. Assigning responsibilities and resourcing training and publishing milestones will turn commitments into outcomes. And here's a practical split you can act on immediately. For scientists, you can enroll team members in non-animal training, reserve time in a non-animal core facility, and when appropriate, submit evidence packages that rely only on non-animal methods. Anchor your submissions on OECD test guidelines and in a clearly defined context of use, with an explicit statement of uncertainty, and switch to animal-free media and reagents and report performance in relation to the decisions you are informing. And for regulators, you can schedule continuing professional development on weight of evidence, integration, uncertainty analysis, and next generation risk assessment. Pilot joint reviews with applicants using real cases. You can also adopt reviewer checklists that discourage reflex requests for animal data, and track gains such as time to decision consistency, and avoided animal use. We have seen why the status quo underperforms, where policy is opening doors, and how training standards and smart incentives remove friction. The path forward is both ethical and practical. Better science, better decisions, and fewer animals. Let us shift the mindset, build the skills, and align the rules so that the ethical choice becomes the scientific default and the regulatory standard. Thank you for your attention. And if you want to stay informed about training opportunities and workshops and funding, please sign up for our Center for Alternatives to Animal Testing newsletter. And I'm happy to follow up by email as well. Thanks again, and I'm looking forward to your questions.
Thank you so much, Dr. Katrin, for your awesome presentation about all the ethical standards and all the issues that the community is currently facing in relation to the animal-free innovation use. Thank you so much for your presentation. I hope that everyone can have some questions and can pose them in our Q&A box for Dr. Katrin. Now we can move further, and Professor Emanuela Corsini will present her slides.
Thank you so much. I hope you can see and hear me well. Good morning, good afternoon, good evening, because I guess that we are connected in different regions of the world. Now, I have been asked to provide you a sort of an update on the approaches, the 3R, to skin and photosensitization. Okay, so then I will try to keep the time, because we can absolutely say that the skin sensitization is a story of a big success for the replacement of animals. So really big things have been achieved. Now, let's see where I can move to the next one. So as a layout of my presentation, I will briefly start with framing the problem, why we need to assess for skin sensitization, definition of hypersensitivity and contact allergy, the concept of the AOP and its application for the skin sensitization. Just to remind you all, the first AOP that has been formally accepted is actually for the skin sensitization. Some examples, I will not be able to cover the different key events, just a flavor. photosensitization, and then I will draw some conclusion. Framing the problem. Now, when we think of allergic reaction, we know that the contact hypersensitivity and respiratory sensitization are the two most frequent manifestations of chemical-induced allergy. It has been estimated that there are many EPI studies. For example, in Europe, approximately 20% of the population suffer from contact allergy. We have a long list of low molecular weight chemicals that are actually positive for skin sensitization. Slightly less is the figure that we have for photoallergy. So for what we know, in patients undergoing photopatch testing, less than 10%, between 4 and 80%, is actually the photoreaction on an immunological underlying mechanism. Clearly, the frequency, the high prevalence also among chemicals, of course, ask for the toxicology to answer the question of the intrinsic contact allergic potential of the xenobiotic. And so based on that, all chemicals need to be tested for the contact allergy potential. So this is the frame. So this is why contact allergy is so important. Now, what is from a mechanistic point of view a contact allergy? Chemical allergy is an immunological specific immune response caused by the exposure to low molecular weight chemicals. And with low molecular weight, I mean chemicals typically less than 500 Dalton. Allergic contact dermatitis is a type 4 hypersensitivity, so is a delayed type hypersensitivity reaction. The C as an effector cells, the cytotoxic T cells, and of course the cooperation with the T helper cells. And is due to more than one exposure to the chemicals through our skin. Over the years, we got quite a good understanding of both the induction and the elicitation phase. And we know that the chemicals need to penetrate the skin when the epidermis, they need to go through the stratum corneum, they need to be then to activate a local reaction with the involvement of the keratinocytes and the dendritic cells. And then we have the migration of the specific or the dendritic cells that bring the potential new allergens to the draining lymph node where the activation of the specific immune response took place. So we have a good understanding of the process, and thanks for the understanding of the biology is that we could actually design the adverse outcome pathway for the skin sensitization. So historically, contact allergen has been evaluated using animals, the oldest one, the guinea pig models, the relatively new, the local lymph node assay are all based on animals. As it was very nicely pointed out by the previous speaker, there is a ban in Europe, but not only on the use of animal to test cosmetic ingredients for their safety, including skin sensitization. And we know that in this context of the in vitro, the application of the 3R, mainly the replacement, is based on the concept of the AOP. And as I already mentioned, the first AOP, it was the adverse outcome pathway for skin sensitization initiating by covalent binding to protein, was the first AOP that was published back more than 10 years ago. So really is a long time. How it looked like this AOP. I know that this slide is quite busy, so we have in the back on top the AOP from the molecular initiating event to the organism response, which is the allergic contact dermatitis. Thanks to understanding of the biological process, we know what is the key event, the molecular initiating event, the covalent binding to protein, which is key event 1. Then we have the keratinocyte, the dendritic cell activation that are called key event 2, key event 3, and then we have the migration in the organ response in the draining lymph node, where we have the clonal expansion, and then we have the apical, the adverse outcome. In these slides, I have also reported the different OECD test guidelines, the different number that they cover the different key event. Of course, the key event 1, 2, and 3 are based on replacement of animals, while for the key event 4 is the local lymph node assay, and the animal models or the human models are the one, the guinea pig are the one that address the allergic contact dermatitis. At the bottom, you see many acronyms for the different in vitro methods, the majority of which has been formally validated, and these are the ones that we can find in the different OECD test guidelines. As you see here, since the molecular initiating event is an electrophilic reaction between the allergen, which is an electrophilic in nature or following biological or following by activation, because we can also have the preaptens, which can be simply by exposure to air, they can become reactive compounds. So, and is binding to the nucleophilic side of the proteins. So we also have in silico models that are also integrating in the defined approach that was mentioned in the previous presentation to come out with an answer. Okay, so many essays. Now, just to give you a flavor on some of these in vitro methods, some of the new one, I will show you one of the latest methods that we have that is related to the keratinocyte activation, because one of the first step, maybe something happened maybe like in parallel with the dendritic cell activation, but definitely the covalent bindings of the allergens to the protein trigger the activation of the keratinocytes. Now, the old one that I want simply to report to you is the EpiSense A, which is based on the use of reconstitute human epidermis, which is a sort of next step, getting more closer to the real exposure, because I mean the methods, the reconstitute human epidermis, you see, closely resemble the human epidermis. EpiSense A is based on the use of lab site AP model, and following 6 hours of exposure, then you go to evaluate by RT-PCR the expression of 4 genes that has been shown to be selectively upregulated by contact allergens. The different genes has a different, for the prediction model, they have a different fold of induction to call your chemical as a sensitizer. For example, interleukin 8, you need to have an upregulation of more than 4-fold. Then the classification, if your chemical trigger at least one of these genes above the cutoff, then you classify your chemical as a sensitizer. In terms of accuracy, correct prediction of positive and negative, you see we have 82% of accuracy. If we move to key event no, not here, so sorry, here. We move to the next key event, which is the dendritic cell activation, because then there is a crosstalk between the keratinocytes and the dendritic cells, and the signal from the keratinocytes are actually required for the migration of the dendritic cells from the skin to the draining lymph node. One of the last essay covering key event 3 is the GARD skin. The GARD skin is actually the first methods based on toxicogenomic approach. So they run a wide RNA array, and then the prediction is based on the gene expression of 200 genes known to be relevant for the skin sensitization. It's based on the use of a surrogate of dendritic cell line that is called MUTZ-3. And now, I mean, of course, then the company that own the patent for this assay, so then as you have to buy the cells from them. And if you look at the overall accuracy is 83, so similar to the one that I show you for the EpiSense A. And we also know that there are several other methods that are waiting for validation and acceptance. Because, I mean, it's important to have more than one to cover, not really to cover the different key events, because we have several already for the different key events, but the applicability domain is not equal for the different assays. Okay, so, and there is no a single assay that, I mean, unless you go for a positive classification, other way than you need to combine. So what we have achieved for skin sensitization, we have several validated methods for the hazard identification, yes, no. We have a defined approach for the subclassification accordingly to the global harmonization system in 1A and 1B, but we can also use the same validated methods that offer a concentration-dependent response to derive a point of departure that we need for quantitative risk assessment. And one of these approaches, which is based on a Bayesian or a probabilistic approach, has now also been added to the OECD test guideline 497 that was already mentioned before. So once the regulators will accept the use of an alternative approach for a quantitative risk assessment, which is not really formally needed from a regulatory standpoint, because we need for classification 1A and 1B, in reality, with this new approach for the point of departure, we do not really need additional animal testing. So I will skip the concept of the defined approach, because I don't think this has been a part of the OECD test guidelines. Let me see, because I thought that I have. Okay, so while maybe I just want to mention that beside the Bayesian approach, the SARA-ICE, which is an online tool based on you need to input, there is an an Excel file in which you need to upload the information from the keratinosense, the HGLAT, the physical chemical characteristic of your chemicals. Then you fill the file, and then you load again online, and then you receive, I mean, you got a base, so that the system will elaborate the data and come and provide to you the classification, including a point of departure. But there are also other methods to derive the point of departure, maybe more straightforward and easy, such as the one proposed by Givaudan with the deterministic regression models. And here is the link. So it's an equation. Also there you need to input the results from the in vitro test, and then you have an estimation of the no expected sensitization induction level. Similarly, published last year based on the GARD, not the GARD skin, the one is in the OECD test guidelines, but the one that include a dose response. You can also do an interpolation more than extrapolation, bilinear regression, where that allowed you to estimate the point of departure. So they are both, in a sense, easy system and transparent system to be used. And so really, which is quite important to make skin allergy a sort of like a memory of the past. So if you really want to try to replace, I mean, not to fulfill the initial duties of trying to reduce the incidence of skin sensitization. Now I will spend a couple of minutes for the photosensitization. We'll say like less than 10% of the phototoxic compounds, actually the phototoxicity as an underlying mechanism, the induction of a specific immune response. Photoallergy as contact allergy is also a type 4 hypersensitivity. Of course, the interaction between the UV light with the chemical that resulting in an excitation of our chemicals, we see quite often also the generation of a reactive oxygen species that can trigger the phototoxicity, which is a very broad name, under which we also we should distinguish between photoirritation, where the majority of chemicals that are photoactivated actually induce photoirritation. Second, we have photoallergy, and only few chemicals can induce photogenotoxicity. Okay, here is, I know that is sort of busy slides with many information. The take-home message is that some of the methods that I show you before that has been formally validated has also been adapted for the classification, for the characterization of the photosensitization potential of xenobiotic, because you see the DPRA, the photodipra, the ADRA for the key event 1. We have these are based on methods that has not been yet formally validated, based on the use of interleukin 18. Then we have some of the dendritic cells methods, and then we have the GARD skin and the GARD skin dose response, which basically, so you use the same strategy with the addition of the UV irradiation. You see here the predictive capacity in terms of sensitivity and specificity, the overall accuracy. And you see, for many, we don't really have the number available, and only a limited data set in terms of chemical that has been really tested. The bottom line, and there is a literature that show the optimization of the methods to detect skin photo sensitizer, but none of these methods has been validated really to prove that the methods is good enough. Coming to the conclusion, so as I told you, we have several in vitro methods to assess contact hypersensitivity. More than 10 methods has been successfully validated, and their integration allow appropriate classification and labeling, including the possibility to derive a point of departure for quantitative risk assessment. Concerning the photosensitization, the validation and acceptance of such methods is still pending. With that, I thank you so much for your attention, and also I would be glad to further discuss at the end of our presentation. Thank you so much.
Thank you very much, Dr. Corsini. Unfortunately, we haven't presented you before. I will say that she's full professor of toxicology at the University of Milan, and her research is focused on refining alternative in vitro immunotoxicity. She has a lot of experience in scientific organization based in Europe, ISA, EMA, the CCS, and also international, such as the OECD. Now, thank you very much, Manuela, for your nice presentation. And I have a question in the Q box, and then at the end we will read all the questions for all the presenters. Thank you. Now our last speaker is Dr. Thomas Hartung. He's one of the pioneering alternative testing methods. He was the head of the European Center of Validation of Alternative Methods many years ago. And he's now the director of the Center for Alternative to Animal Testing, CAT, in the US and also in Europe. He has a lot of papers, more than 700 papers, and a lot of H index, very high. And he is also professor at the Johns Hopkins Bromberg School of Public Health. There is a lot of places he's teaching, and also he teaches in the Coursera toxicology classes with a lot of students for this class. Thank you very much, Thomas, for accepting participate in this webinar. And his talk is Alternatives to Animal Testing, see what the ugly duckling has become. Thank you, Thomas, again.
Yeah, thanks for having me and for the nice introduction. It's my pleasure to talk, and one of the big messages here is really things are changing. We are changing from a discussion which was very much driven by ethics for many decades, and where it seemed to be just a good thing to reduce animal suffering, to something where we increasingly accept that there is scientific reasons for change and there's economic reasons for change. And I would like to touch on all of this. And I would like to show you how the ugly duckling has become a beautiful swan, how the early alternative methods with all of their shortcomings are now shining as new approach methods, as microphysiological systems and AI. Made my slides available. You see the QR code. I also have the same QR code again on the last slide. So if you're not fast enough to grab your phone, that's the fastest way to do. I'm very much interested in evidence-based approaches. Some 20 years ago, we started this with Sebastian Hoffmann. And we decided we need something in toxicology similar to evidence-based medicine and clinical medicine, where objective tools like systematic reviews help us to move, because we need to get out of this war of faith, where some people like animal tests, others don't, and some are in between and say, yeah, but it's a necessary evil. Let's objectively discuss these things. And starting in ECVAM in 2007, we organized the first conference. Since 2009, some almost 17 years now, I have a chair for evidence-based toxicology in Johns Hopkins. And we created in 2011 the evidence-based toxicology collaboration, which has become, over time, interacting with more than 1,000 people, a driving force for systematic reviews in our field, which you can see here. And in 2023, we transformed this to a membership-driven society with its own journal. And it is free to become a member. And actually on the 24th of this month, we will have our virtual General Assembly. So if you're interested, please go to the website. And if you want any more details, this is an article from last year, which summarizes the experiences on the road and is describing where we are heading in this field. So I think we are advancing. You see my quest here. I want that the red is no longer the golden red around which we dance, as a lot of toxicology is still doing, but it is more or less starting to get retired. I invite you to read this article, which is written for lay audiences, but is summarizing, I think, quite nicely the transition with new tools where we are moving. And this is, on the one hand, from 2D culture to microphysiological systems. Again, a review article from this year here, and it is from this early computational approaches which had limited applicability domains to something which is at the moment, because of the advent of artificial intelligence, really getting us into a different way of doing toxicology. And I will address these issues. But first of all, it should be clear, society wants us to change. About 50% of Americans, up about 15% over the last 20 years, don't want animal experiments for medical research, and not for cosmetics, for medical research. 60% of Europeans say so. And the numbers are about 20% higher if you ask for cosmetics. Politicians listen to what their voters want. And you see here in black noteworthy European legislations and the reactions of the US, where in 2016, the Lautenberg Chemical Safety Act, also known as the revision of the Toxic Substance Control Act, was bipartisan legislation, which was introducing alternative approaches and forces the EPA to reconsider. But also the FDA Modernization Act from 2022, which was unanimous, did take up, as Katrin very nicely said already, did urge the FDA to consider novel methodologies. At the same time, we see the difficulties the animal tests have been creating. We have chemophobia. More people are fearing chemicals and their health. And the assays are imperfect. Take the cancer assay as one of the key concerns of a general audience. It takes you 4 to 5 years to get the result of this 2-year animal test, because it takes about 2 years to do all of the histopathology of 400 animals and all organs. It does cost up to $1 million for one assay, uses 400 rats, and you need 10 kilograms of the substance to feed on a daily basis 400 rats, is whatever you're testing. Tell a chemist to produce 10 kilograms of something which is not yet in use. And there's a lot of shortcomings of this assay. Of all the 3,500 assays which are in the public literature, 53% were positive. Sure, we tested some which did smell like problem, but the estimate is still that only 5 to 10% should have been positive. Between the assays themselves are only about 60% reproducible. For some 120 assays which have been redone. And the correlation between mice and rats is only 57%. None of them is likely to predict humans any better than they predict each other. And it's very different organs which are affected. The potency of substances varies. So it is really a very limited tool. I have been collecting studies where hundreds of chemicals have been tested for different aspects of their accuracy, whether it is reproducibility, whether it is interspecies effects, whether it is prediction of humans. You can see at best they are 80% accurate. This is the quality of these animal tests. And this is opening up increasingly for something better, because pharma is noticing that they are spending a lot of time showing that certain toxicological findings in animals are not relevant so that they can proceed with their clinical studies. You heard already that the times are changing. We had a miracle April this year, where the FDA on the 10th of April announced a roadmap to reduce animal testing. At the same day, the EPA renewed their pledge to move out of animal testing in 2035 as a standard requirement. The pledge was done in 2019 by Andrew Wheeler already, and only some 19 days later, NIH released a press release to prioritize now human-based research technologies such as organ-on-chip systems. And also in the EU, on the 23rd of May, the Council endorsed the European Research Area, and one of the few things which were named are alternatives to animal testing. And you heard already about the roadmap coming up early next year after several years of discussion. The QR codes lead you to two of my comments which I published on these most recent developments. So what we are seeing at the moment is a transition, a change of technologies taking place. And in this article from April, I compare this to 110 years ago when we were started to change from another animal technology, which is horse coaches, to cars. And it is funny to read how much the discussion at the time resembles the arguments we hear now, how reliable this technology is, how long we have built experience in doing it. But only the availability of an infrastructure and more experiences of use led to change that some decades later it seemed laughable to defend the horse coach against the car. And I hope that something similar is taking place here. It is really about integrating here, shown for the drug screening process, microphysiological systems, AI, but also biomarkers. And it is about a combination of this, what Emanuela showed as IATAs, as integrated approaches to testing and assessment by the OECD. In the end, I call it in vivitrosi, in vivo, in vitro, and in silico, reasonably combined. And this is very necessary for the drug development, where 57% of the new drugs at the moment registered are actually biologicals. They're either antibodies against human proteins or structures, or they are human proteins. And they can simply not be tested in animals. And this alone is opening up for the new approach methodologies. One of them being MPS, microphysiological systems, an umbrella term for a lot of different things, from tissue chips to organoids to spheres and organ-on-chip systems. The FDA used this term, and it is becoming increasingly used. We started with a workshop report in 2014 in this field and have been developing this with a number of strategic workshops. We developed Good Cell Culture Practice 2.0 for quality assurance of these systems and started then the MPS World Summit series in 2022. The last one just took place in June in Brussels. And we created an international society to organize these conferences and other activities in the field. You see me here handing over to my wife Lena Smirnova as the president of the society last year. And the society already organized now the Brussels event. And you see how nicely this has increased from 665 people, notably 10% came from the FDA in the first meeting, to now almost 1,500 participants. A very vibrant conference. And the next one will take place at the end of May in Washington. So stay tuned if you're in this field. Our own work on this is in the field of brain organoids. You see this here from induced pluripotent stem cells. They show all type of neurons, oligodendrocytes, astrocytes. We also added most recently microglia as the resident immune cell to the system. And you can use these highly standardized organoids. You see them here. 1 6-well plate gives us about 4,000 of these brain organoids. And they are alike in size and consistent in proportions of different cell types. Very reproducible also from week to week. And as I said, mass produced. We can do them from different genetic backgrounds, or we can modify with CRISPR-Cas and others the background of an individual so that we can modify it. And we have been using this for autism research, COVID-19. We grafted them with glioblastoma, trauma, and others. So we are permanently adding disease conditions to this. Alzheimer, most recently. This is one example. Lena and team were able to find the first pair of a risk gene and a chemical which are producing synergistically features of developmental neurotoxicity, so possibly being a cause of autism. And I think this is our philosophy. We know genes play a role, but we need to find the triggers which are producing the disease when exposed. And based on this, we here in Hopkins received a $12 million grant from the NIH where now in 18 centers, 175,000 children are being monitored for possible interactions of genes and environmental factors. And we use our brain organoids to show whether we can verify such synergies similar to the chlorpyrifos case, a pesticide, which was, by the way, in the meantime banned because of these possible links. Just to show you the most science fiction approach we are using at the moment, we are talking to our brain organoids through microelectrode arrays and combining them with AI. And what we achieve is something which we call organ intelligence. What you can see here, this little jumper here is controlled by AI, by the brain organoid, and it learns to jump. And we are measuring by the distance it achieves on average whether there's a learning success. This is work in progress, but we are increasingly publishing also on the machinery, what we can measure and what the reactions and what the reorganization of these brain organoids is when doing these type of things. The other transformative technology at the moment is AI. We simply see a synergy of different growth curves which are synergizing. Data in the world are doubling at the moment every 18 months. So 90% of all data have been produced in the last 3 years. Our computers are doubling for 60 years, every second year for half the price and their capacity. That's known as Moore's Law. And AI, since deep learning was introduced in 2010, is doubling every 3 months. So this year's AI is 8 times better than last year's. So this is really an enormous growth. And I believe that this is leading us to a different type of doing science. I've laid this out in this article from 2 weeks ago, where I describe how we are moving from science written with IE to science written with AI. And while we're at the moment trying to integrate AI into science, we will increasingly be doing science for AI as the central knowledge integrator, feed and identify the gaps and do experiments to fill these gaps. So what we now need to deploy an AI-driven toxicology is most of all building trust. And just to give you a flavor, this is our current model, which we have developed in the context of the EU OnTox project, a $20 million project with 17 partners. We've created databases to import. Earlier this month, we published on this BioBrick project to import entire databases with one-line commands. And the first 12 of these gave us 117 million chemicals with data. In total, almost 260 million chemical activities where we have a chemical, a property, and a result. And that's a treasure trove for data mining. And we have 4,000 different properties of these, mostly in vitro or chemical-physical properties, for which we have more than 1,000 entries. And these 4,000 properties we can predict in training on average 91% accurately. 91%. That's crazy, isn't it? And I think this is now at the moment in evaluation with the project partners, and we hope to publish this model and give access to the model towards the end of the year, early next year. Now it is really about thinking, how can we use such information which comes by pressing a button. This is an article written with Robert Califf. At the time of writing, he was the head of FDA, head of 19,000 people, and we are discussing how to build trust on these methodologies. And I think that is really now about finding out how bias, uncertainty, probability, and causation are influenced with the new tools of AI, and how we can really build something which is mechanism-driven, where we can estimate our uncertainties and reduce biases. The big picture, we have these disruptive technologies, and we also have some exposure science opportunities. We can measure more and more with more and more sensitive things with sensor technologies. And this is feeding into big data, into AI. And this is first of all giving us the opportunity to help chemists. We are always thinking of regulatory toxicology, but the same tools allow us to either design chemicals which are safe, benign by design, if you predict computationally, You can do this before you synthesize, and the chemist can choose the safer chemical as an option. Or we can test early when we only have small amounts of a substance in the in vitro systems and can help to decide what to put your money on and what not. So this is called green toxicology as a core part of green chemistry. And then combining with exposure sciences, we can start what I like to call exposomal intelligence, EI, in order to understand what are typical or what are correlations between exposure and certain diseases. And can move it into digital twins and other type of assessments and start working on the human exposome. And also opening up these digital twins are also the tools for personalized medicine. Our textbooks are full of advice for the 70-kilogram male, and we have now more and more patients which are 70 and have survived one cancer and are diabetic and obese. And this is something where we need to personalize, and this is where digital twins come in. So it's about safer products, better treatments, and safer people. And this is all based on a certain set of technologies and AI. So to say a few words about these, green toxicology is a program we started in 2014. Alex Mertens, assistant professor on my team, has the lead here. She just published 2 years ago this first textbook on green toxicology, and it's really about the green toxicology principles of green chemistry. Just in May this year, I was part of a group of 30 people through a Nobel Symposium led by Paul Anastas, one of the fathers of green chemistry. We came to the Stockholm Declaration towards Chemistry for the Future, which is all embracing these technologies at the moment. On the exposome side, Fenassi in my team, also assistant professor and deputy director of our center, just organized in May an exposome moonshot forum. This is the idea. We need to get started to develop something to complement the human genome with a human exposome, so the exposure side of disease. And we invited 300 people to Washington on invitation only and brought together opinion leaders in the science with the civil society, with policymakers, founders, and to find out, are we there? And a critical role is here again with AI and exposome intelligence. I just wrote an article on this in Nature Medicine, so if you're interested, give this a look, please. The forum ended with a declaration towards the establishment of a global consortium for the human exposome, and we're now establishing work groups, newsletter. Still possible to sign this declaration to support this and become part of it. Does not cost anything, but you get informed and can be part of the working groups. With this, I would like to close. I believe it's now or never. There's a lot of signals of green. We can integrate these disruptive technologies with knowledge by AI, and they come from the alternatives to animal testing field. And we can, doing so, accelerate product development. We see already how AI and others are impacting on drug development. And I believe that we also can optimize prevention, public health, and environmental health, and democratize healthcare access. As I said, the slides are available, and I hope we can now start discussing.
Thank you very much, Thomas, for sharing your presentation and your articles. This is very helpful for everybody. Now we have finished all the presentation, and it's the time for questions. Here in the question and answer box, there is one question. This was answered by Emanuela Corsini, but I will read it for all the people. Nobody, we don't know who is him or her. This question is for Professor Emanuela. For the skin sensitization KFN2, now the EpiSense A has been adopted by OECD and uses tissue models that are closer to human. What is your perspective on non-concordant results between the EpiSense A and keratinosense? In other words, could the EpiSense A results should be considered to have more weight in a regulatory context?
Okay, so I provide an answer, a sort of explanation on why the two models addressing a key event 2 may actually provide at the end not concordant results. Now, this is not surprising for the reason that I put on as I comment as an answer to the question. First of all, of course, I already thank for the nice question, and definitely, I mean, is a point. We also note that very similar, like if you compare the Lucens with the what is the name of the other that is based on the Lucens with the HGLAT, where they both look, they have in common CD86 upregulation. They also, they are not the way of exposure, then there are chemicals that are not concordant in terms of positive or negative classification. So definitely the same could apply for reconstitute human epidermis and the keratinosense, which is based on a human cell line. Okay, so then, of course, I mean, so different target here, because one is based on the addressing the upregulation of genes involved in the oxidative stress. The other one that maybe also are linked to the oxidative stress, but they also can be triggered by different signal transduction pathway, which means they do not really fully overlap in terms of underlying mechanism that they target. Okay, so these are the other difference is the bioavailability, because I mean, traditional cell line, what you add in the culture medium, some will reach the cells, while once we use a reconstitute human epidermis, then the chemical has to go through the epidermis, reach the viable epidermis. So this could also explain not consistency in the positive or for some chemicals, only for some. So these are the one I love, the really two possible explanations that come to my mind. In terms of what could be the regulatory acceptance, since the reconstituted epidermis is more close to the skin, would the regulators give a more weight? These need to be seen, okay, with more chemicals, once we have a better understanding on the applicability domain, so then we may be sure that if we have, because I mean, the concern is a false negative, okay? So this is the big concern. If you have a false positive, of course, from an industry standpoint, it's not so good, but at least we are not missing chemicals. So I would say, and the other thing would be to see implementing these methods, for example, in SARA eyes, how this will change the point of departure. So then this is also important, and similar for the defined approach. So we have to see once all this has been like fully accepted in the defined approach, so then we may decide to give a different weight, more for the reconstitute-based methods. I hope that I address your question and concern.
Well, there are no more questions. This may be good or what? And then I want to make a question to Professor Hartung. Was related to the application of the artificial intelligence to this kind of studies. But as you pointed, there are I suppose there are difficulties for implementation this in the regulation, because we have problems with a lot of studies in vitro or something like this to be validated and to be accepted. And what is your opinion about the artificial intelligence? How many years do you think would happen this acceptance by the regulators?
I mean, from my point of view, every year waited is a year wasted, because these methods are already delivering now. We have only to ask what they deliver. For example, they are helping us to find literature and other information on a given substance without then doing anything else. But if you just think in the life sciences, 3.4 million papers are produced every year. PubMed in 2023 alone took up 2.2 million new references. 2.2 million, yeah? Nobody can read this. And AI is better in annotating scientific papers than humans. And it does not forget. And sure, if you go for generative AI, there's hallucinations, but this problem is coming under control. Hallucinations, so inventing references which do not exist, was about 9% last year. This year it is between 1% and 3%, and ChatGPT-5, for example, says it is at 0.7% at the moment. I do hallucinate more than 0.7%. So it is really getting better in this respect too. So there's a lot of uses now, even for imperfect AI. We will never be able to assess all of the unknown substances which have never been tested. A study from 2019 shows 350,000 chemicals registered in the 19 most developed countries for the commercial use. 350,000. We have data for some 30,000 probably with REACH now, but not much more. Very rarely comprehensive. We have situations where we just want to do a plausibility check. Yeah? There's this wonderful dossier which says no problem at all, fit. Yeah, why not check it with an AI tool and ask the submitter? We have emergency situations. There's a spill, no information available on the substance. How can we quickly get something to find? Yeah, so there's already now uses for this, and we are stupid not to employ it. But the good thing is everybody sees the urgency. Everybody sees that something big is happening, and I'm talking to many of these agencies at the moment about training of their staff on what to do and how to get carefully, not rushing it, but a good path forward to implementing it.
And the problem is there are still reluctance for this application, but as you say, more publication are needed to convince people that this is a good way to obtain results. Another question, this is for Dr. Katrin Herrmann. As you, in your presentation, you put something about the barriers to adoption of NAMs. This is really a problem, these barriers, especially in the journals, so more than in journals for the reviewers, because the people who, as me, working in alternative methods, many times the reviewers say, hey, okay, it's good, but you have to do experiments with animals to confirm your results. And this is an important barrier. And many people sometimes perform this study to publish their results. But if we don't want to do this kind of experiment with animals, what would be the situation, the ideal situation, or what will be the recommendation for reviewers and the journals, especially the editors of the journals, to accept NAMs without experiment with animals?
Yeah, thanks for this question. For some reason, I skipped that slide. I had a slide on animal methods, so-called animal methods bias. That's exactly where animal experiments are actually not necessary, but they are still requested by the reviewer or by the editor. And I highly recommend, I can also put that in the chat, for people to check out the CoLab. We have a website, it's called animalmethodsbias.org. It's an international group of people, and we are working on that issue. We actually developed editor and reviewer checklists to avoid this kind of bias. And yeah, I'm sorry that I skipped that. I don't know what happened there. It was actually in my presentation, and it's really important. And maybe to add to this, it's already starts with the funding, but like bias for towards animal methods when you apply for funding. It could also already start in your laboratory if you only have access, or your PI wants you to use the animal models that exist in the lab, then that's already also a problem. But thanks for this question, and I will share the link. And also, if anybody wants to join our efforts, we are happy about this.
Yeah, you can share the link in the chat.
Yeah.
Maybe. Something I think there is another question. Sorry, here. There are more questions. Stefan Grunk say, for industry scientists who are only familiar with traditional animal-based toxicology methods, what could be a good first step to start using NAMs and contribute to their further development and regulatory acceptance? This is an open question for Thomas, for Katrin, for Emanuela. You can answer this.
I mean, for me, the starting point would be to employ more AI, be open to these new methodologies, and ask your favorite chatbot when you come up to something to explain what it is and how the current state of the literature is. They are damn good in assessing and giving and summarizing these things. And most people don't understand that these large language models are chatbots. They're meant to chat with you. So you should not just pose a question and say, okay, no, ask the AI to refine, to apply it to your use case or whatever, and you will get information. Yeah. So something we tried desperately for many decades of providing the information about these methods to people was never tailored to your needs. Now you can get it very quickly. And I would also recommend very much to subscribe to our newsletter, which is free, obviously. Shoot me an email or look on our website, because you get on a weekly basis what's happening in the field, and there's often something you might want to follow up.
Yeah, and if I may add, like also a comment, of course, I'm sure like working for industry, when you want to apply for registration of any new chemicals, of course, I mean, you have to follow what regulators will ask you to do, basically. I mean, it's true that now the interaction is getting more and more they try to reduce as much as possible the use of animals. But nevertheless, there are some animal studies that unfortunately they are still require and ask. Things are changing. We will get there, but maybe not by tomorrow. And of course, any new validated in vitro methods approach in silico then need to be implemented in the different regulation, becoming somehow mandatory, meaning that for a specific endpoint, you are no longer allowed to use animals. Think of in the past for corrosion, skin toxicity, ocular toxicity, more recently for skin sensitization. So we hope that in the future, we will be able to predict the possible any adverse outcome based on alternative approach, and definitely the use of the AI also integrating the different information, I mean, is an incredible tools that we definitely need to take advantage of the new knowledge that we have. Thank you.
Yeah, I can also add something maybe. I mean, I mentioned in my talk, it's very important that the staff is trained properly, and there are NAM modules, and of course you can book time in NAM core units. For example, I mean, this is academia now, but in Berlin, for example, they have a unit where any research group can come in and get hands-on training on organoids and to familiarize yourself with new technologies. And I think that's very important. And I'm sure that especially industry does have such training possibilities, and also core centers. And also maybe for actually for everybody, but if you're based in the US, you could also, of course, travel there. Next summer in June, it's June 15th to 18th, we offer a free summer school on NAMS, animal-free methods. And everybody's welcome. It's free of charge. You have to pay your travels and accommodation, but the program itself is free of charge. And we do that together with the Physicians Committee for Responsible Medicine that actually established this summer school series. And it's happening every other year. Maybe you're aware of the JRC Summer School. That's the equivalent in the US. So I highly recommend coming, and you can also visit our lab and many more things.
Okay. If you want to add something else, it's open. There are no more questions in the chat, in the question. Then only to thank all the speakers for your nice presentation, and also the audience to come here and learn more about alternatives. I think it's very important, the aspect of education, as Katrin said in her presentation, is basic, this education especially for young researchers, because sometimes they only know animals, working with animals, and then don't know there are other alternatives. The people working in alternative, of course, we know all about alternatives. We attend the congresses of alternatives, but there are another scientists that they didn't do this, and then it's difficult to move their mind to alternative methods. And then this kind of webinars and all the activities related to the diffusion of the promotion of alternative methods is a good idea. And it's important that editorials support this kind of activities, of course. Then thank you again, all the people, and I hope this will be helpful for you. And thank you very much again. This is all.
Thank you very much, everyone, for joining us on this webinar. I want to thank all the speakers and also thank our chair, Professor Maria. There was also one more question in the box that was answered by the Professor Thomas, and it was related to the research lab. Professor Thomas has replied that they have 4 postdoc positions for data scientists, bioinformaticians, and so on. So you may also see his reply in the chat box. I would like to thank you all for joining this webinar and being a part of it. The recording will be available on our SciForum page as soon as possible, and also the certifications of attendances will also be available on your profile. So thank you, everyone, and I hope you have a great rest of your day.